In the world of biotherapeutics, ion exchange chromatography is used for charge variant analysis. Laboratories in this field are typically either doing this by ion exchange with HPLC or else are using a capillary electrophoresis technique. But briefly, why is it done? Because variations in the charge profile could mean that there was a modification that occurred during manufacture or storage of the biotherapeutic that could be harmful or even fatal to a patient. So being a bit more specific, charge variant analysis is a separation of proteins based on their accessible surface charge. And we all know that biotherapeutic drugs are made up of large heterogeneous molecules that can be subject to a variety of post-translational modifications, always a big mouthful, during manufacture. And they can be things such as glycosylation and lysine truncation, and of course many more. In addition, chemical modifications can occur during purification and storage, such as oxidation or deamidation, just to name two. Ion exchange charge variant analysis is used to identify some or hopefully all of those modifications. And as most of you will know, charge variant profile is mandated by the regulators and by regulations such as ICH Q6b. A few basic facts about proteins and their charge states. We all know proteins are composed of amino acids, and amino acids are charged species, so proteins are always carrying a charge. And that charge distribution is not uniform. A protein can have both positively and negatively charged patches on its surface, and this is quite nicely illustrated in the image on the right of your screen. The pI, or isoelectric point, is the pH where the net charge of the protein is 0. But of course, there is still charge, as we know, but the net charge, adding all those charges together, is 0. Ion exchange separation is based only on the exposed surface charges of that protein, which will change with the pH. So despite the known pI of a denatured protein, a protein can still interact with an ion exchange column due to pockets of charge at the surface. And this charge state is important for the ion exchange separation and will change with the pH. So it's important to control the pH during the separation to conserve the charge on the protein. And we're going to look at some examples of the importance of this later on in the webinar. So let's talk about what's happening in an ion exchange protein separation. Excuse the rather complex graph on your screen, but it's, to be honest, probably familiar to most of you in the audience anyway. And there are two general mechanisms to retain proteins on and to elute them from ion exchange columns. There's the more traditional salt gradient and what we're going to call the emerging pH gradient. I know it's been around for some time, but it's still not as widely used as salt gradient. So what's the effect of pH on the charge on the protein? It's a really important consideration as the charge varies with the pH and the separation is based on that charge. For reproducible results, the pH and hence the charge on the protein must be carefully controlled. As the pH increases, the charge on a protein goes from basic to increasingly more acidic. At some point, the protein will have a net charge of 0, which, as I mentioned, is the isoelectric point. At pH values below this, the protein has a positive net charge, and above it, the protein has a net negative charge. In salt gradient-based IEC, the pH of the buffer system is fixed and must be controlled. In addition to choosing the appropriate pH of the starting buffer, its ionic strength is kept low since the affinity of proteins for IEC resins decreases as ionic strength increases. Proteins here are eluted by increasing the ionic strength, which corresponds to the salt concentration of the buffer, to increase the competition between the buffer ions and proteins for charged groups on the IEC resin. In this way, the interaction between the IEC resin and proteins is reduced, thereby causing the proteins to elute. So contrast that with pH gradient-based IEC, the pH of the starting buffer is kept such that the protein obtains the opposite charge of the stationary phase, and so binds to the stationary phase. The proteins are then eluted by changing the buffer pH, so the protein transitions to a net 0 charge, ultimately the same charge as the resin, and is eluted from the column. One of the benefits of pH gradient-based IEC is that the salt concentration can be kept low, yielding less buffer interferences. So basically, it's a story of attraction. Ion exchange chromatography separates proteins and other molecules in a solution based on differences in the net charge. Negatively charged molecules bind to positively charged solid supports, and of course, vice versa. In principle, a protein could bind to either a cation or anion exchange resin, but in practice, proteins are only stable within a narrow pH range, and the choice of resin depends on the pH stability of the protein. Ion exchange resins are characterized as weak or strong exchangers, and you should note that the terms strong and weak refer to the ionization rather than the strength of the binding. So in these terms, SCX refers to strong cation exchange, and WCX obviously refers to weak cation exchange. These terms refer to the extent that the ionization state of the functional groups vary with pH. A weak exchanger is ionized only over a limited pH range. A strong exchanger shows no variation in ion exchange capacity with changes in pH. And as we'll see, the two types can offer complementary selectivity. But I find you asking, is it as simple as that? For example, if I know that my protein has a pI greater than 6, what do I try first, WCX or SCX? Well, actually, in fact, both could work. They're complementary, and we'll have selectivity differences so that one may work better on one protein and the other better on another. You can think about this a bit like selecting a C18 column. How do you know which C18 column to use out of the many that are out there? Well, some of it comes down to personal preference, your own experience. Some will be a guess, and some will be based around what you read in the literature. So really, you need to test both chemistries to understand which will offer better resolution for a particular protein. Whether using a cation or anion exchange column, sodium chloride can elute the bound protein. In anion exchange, chloride is the counterion. Chloride is exchanged for the target protein, which of course is then released. In cation application, as shown on screen here, the counterion is sodium because the sodium cation is exchanged for the target protein. So you see a very small animation on screen there, and in that animation you saw the sodium cations preferentially bind to the resin and thereby cause the proteins to elute. So let's take a bit more of a look considering some of those different chemistries. Weak exchangers can cause ionization and therefore change the charge to have an inherent buffering capacity. Strong exchangers don't do this. And of course, selectivity can be different for each ion exchange type. The smaller particle size with more of the recent column types typically have higher efficiency and higher resolution, and we'll take a look at an example of that a little further on. By improving the column chemistry, the lot-to-lot variability can be reduced. It's also important to control the linker length holding the active charged groups. Considering our own columns, we observe similar selectivity for the ProPak Elite, whereas the MAPPAC SCX offers different selectivity. And here's an example of that complementary selectivity. On screen, you can see chromatograms of the same biotherapeutic using WCX and SCX 5 micron columns. One is a strong and the other a weak cation exchanger. You can see where the arrow indicates where there has been a selectivity change. And so just to repeat the message that's here, the choice of column to use will depend on which column offers the best selectivity for your protein. And really, there's no substitute for experimentation. It's also briefly mentioning that the UHPLC system that you use for charge variant analysis should be inert to run the corrosive salt buffers used in the salt gradient elution. I'll be focusing on the use of pH and conductivity detection for method optimization. This technique is extremely useful to ensure the method has good control of the pH and that the column has enough time to equilibrate for the next run. Running blind to the pH profile can often lead to poor equilibration at the end of the chromatography run, which then leads to an unstable method. And we're going to show you some pretty clear and obvious examples of this later on in the webinar. Okay, so let's now consider some specifics. Column length is obviously going to be an important factor. And here on screen, you can see 3 chromatograms of a biosimilar of trastuzumab with column length increasing from 50 millimeters to 250 millimeters. The increased length does offer more resolution. You can see that from the chromatograms, but it's a trade-off you might be willing to consider because of the potential time saving of a shorter column. And what about the thorny subject of buffers? Buffer design for charge variant analysis is very important to keep control of the pH and thereby keep the protein in the same charge state that's optimum for separation. This is also essential for method robustness and transfer of the method to other laboratories. So we recommend neutral goods buffers, which can interact right to the column surface on cation or anion exchange resins, controlling the pH throughout the column. And there are a few examples on screen. The acronyms all refer to specific buffer types, and I've shown MES there on screen. If you're interested in learning what the full chemical names are for each of the buffer types, please do contact us for that information. I wanted to just make a quick note about phosphate buffers. They were recommended many years ago and were actually used in the original documentation from one of our historically very significant columns, the Propac WCX10. But what about now? Actually, the use of online pH monitoring and increased knowledge has made us realize that these are not really good buffers to use. They carry a strong negative charge, and so does the exchange column, and so the phosphate is repelled from the column, meaning the pH might not be controlled adequately. So we wouldn't recommend using these buffers from now on. Luckily, commercial buffers are available, which can be used for pH gradient elution. However, they can also be used very successfully to control the pH during salt gradient elution as well. The image you can see on your screen is of our own CX1 buffers. And these are a strong zwitterionic type buffer mixture made up from a selection of goods buffers, which control the pH well from pH 5.6 to pH 10.2. So moving along in complexity a bit, let's now take a quick look at choosing buffers for a universal IEC setup. Using a quaternary gradient pump, the 4 elements shown on screen can be used for SEC, salt gradient elution for anion or cation columns, and also for pH gradient elution for cations. The ratio of the elements C and D to the right of the screen at a total of 10% will determine and control the pH. The element B percentage will be the salt gradient. If using pH elution, then element B will be unused and the pH gradient is formed from the ratio of C and D. And this setup controls the pH over a wide range, and so it can be used to automate method optimization. To be fair, this is quite a lot to take in, so we'll be taking a bit more of a look at some of the details on this slide later on in subsequent slides. So here's a nice example of that. On this slide, you can see that the mixing of C and D controls the pH really well. The measured pH, which is the black trace going through the center of the screen, is very close to the theoretical pH using the ratio of C and D to control the pH. And you can also see this summarized in the small table on the right-hand side. Note that this data was produced without a column in line. The effect of the column on the actual pH produced is normally a forgotten factor, but it does have a significant effect, as we'll see. And remember, the salt will alter the pH of the buffered solution. So I mentioned in that slide about the effect that the column can have. So what's the effect if you do have a column in line? Well, on this slide, you can see that from the online pH measurement, pH is controlled fairly well, but does change even with the strong buffering capabilities of the CX1 buffer solutions I mentioned earlier. So the chosen pH drops slightly during the salt gradient, which can be seen to have an effect just before 4 minutes. However, the pH is fairly constant and reasonably well controlled throughout the run. And using that universal element system we described, you can prepare methods using and changing the ratios of C and D to change the pH of each run. And here you can see traces taken with a variety of different pH values. And for this particular monoclonal antibody, pH 8 seems to give the best resolution of the variants present in the sample. An optimization of the salt gradient can then similarly be done automatically once you've chosen to use your pH value. So the method we've been describing previously uses buffer blending with a strong buffer that controls the pH over a wide range. But actually, most methods published use only one buffer to cover the whole of the desired pH range. And here's an example of buffer blending using a buffer for pH 6. So what could go wrong, you might ask yourself? Well, remember that salt can have a dramatic effect on the pH of a buffered solution. And this is greater when the buffer concentration is reduced. So at 5 millimolar concentration, which is the black trace on your screen, the buffer starts at pH 6, but during the salt gradient, the pH drops quite significantly to almost 4.5, a really seriously big difference in pH. So this buffer is just not strong enough to control that pH. And notice there's also a huge jump back up to pH 6 at about 22 minutes. So that's highlighted on your screen as well. And this is a good measure of the exact column capacity and will change from column to column. In this case, it is after the salt gradient where most of the proteins will elute due to the salt. But when using 10 to 20 millimolar buffer, this jump in pH happens during the salt gradient. It will force immediate elution of any proteins coming out in this area and create a method that will be very difficult to duplicate. So you need more than 20 millimolar of buffer to be in more control of the chromatography. And back to what I said about pH monitoring, if you weren't monitoring the pH, this would not be apparent to you. So on this slide, we have a nice example of just how unstable such a method would be. Two columns from different batches, which have passed all of the column tests and would be fine with any stable chromatography method, are shown. And due to slight changes in the ion exchange capacity, the position of the pH inflection back to pH 6 is in a different position from column to column. Notice also from this slide how much difference with and without the column makes to the actual pH profile. This is due to the buffering effect of the WCX column used here, as I mentioned a few slides back. So the peaks from those same two identical columns are seen to be quite different on this slide, eluting very differently from both columns, which are both good columns, remember. So the method's the problem here, not the column, because the method has no control over the pH and therefore no control over the charge on the protein. So clearly the two columns are giving us a completely different answer, so this would be impossible for you to reproduce. So let's just take stock. What have we learned about the buffers? Well, the buffer choice is very important, and as we've already said, we recommend the zwitterionic buffers such as CX1. And it's important to use buffers in their correct buffering zones. It's important to remember that buffer blending with salt does not work to create the correct pH. And ideally, you should use pH monitoring if possible after the column during method development. And of course, the column type does make a difference. WCX and SCX columns can have different selectivities, and the individual column capacity we've shown can make a difference too. So now we're going to look a bit more closely at the buffers. So looking at buffers for pH gradient elution, the pH must be controlled over a wide range, and so several buffers must be combined to cover the required pH range. Thermoscientific CX1 buffers are made up from neutral buffers that interact right up to the column surface. On the right-hand side, we show some alternatives. Between them, they do cover most of the pH range with some small gaps, and we'll be comparing them to the CX1 buffers in the next few slides. Now, these alternative buffers are made up of a mixture of neutral, positive, and negatively charged buffers, which will not interact at the column surface very well. And they don't buffer the SCX stationary phase very well either. So let's take a look at what's actually happening at the column surface. Typically, a cation exchange stationary phase is negatively charged, either a carboxylic group or sulfonic group. The CX1 buffer employs 4 zwitterionic buffer salts. They don't bind to the stationary phase, nor are they repelled by the stationary phase. So in the alternative buffer systems we talked about in the previous slide, some components will bind to the stationary phase because they are positively charged when pH is below their pKa value, while others are repelled by the stationary phase. What this means is that the true pH equilibration throughout the column can be compromised. Now, without a column in line, both the mixed charge buffers and the CX1 buffer cocktails from the previous slide seem to work quite well, even though you can see that the CX1 buffers are a little bit more linear. But both appear to have really quite a linear gradient. But obviously, you can't run chromatography without a column. And on the next slide, you can see the equivalent traces, but with a column in line. And this really highlights the problem with the buffer using mixed charge components. You can see this obviously when the column's in line. Here, the column is acting as a buffer, and the charge buffers are not interacting with the column surface to allow pH control. The CX1 buffer works fine and does keep the expected linear response. But the other buffer system is not in control, and again will give slightly different results from one column to another. Method development here will be more difficult due to the programmed gradients not being reproduced in the column. And the important point is that mixed charge buffers do not effectively buffer on the stationary phase due to the components of their mixture. And actually, there's more. Another thing to watch is the UV absorbance of the buffer components. Some of these buffer components increase the UV signal during the gradient, reducing signal to noise and making integration difficult on a sloping baseline. What's important here is to be very careful of the buffers you choose and be aware of the effect that the column and salt has on the pH. Buffer blending using pH tables and algorithms to calculate the pH from the base and acid salts will not work when salt is introduced. Okay, so before we move on to the pH gradients, let's compare the mechanisms in more detail. So we start with salt gradient analysis, the most widely used method, requiring high salt concentrations. Proteins are pushed down the column from site to site by the salt ions in the salt gradient. The salt disrupts the protein to solid phase interactions, and a focusing effect is seen as the salt level increases. So the effective length of the column is important. Typically, a long column and long run times are required. But careful control of the buffer is needed. For example, if there is high salt present in the sample, it will act as an eluent to prevent the protein binding to the column until buffer exchange takes place. This means that the injected sample has not bound to the top of the column as normal, but has gone through the first region of chromatography without interaction with the resin, and so changing the effective length of the column. In an extreme case, it elutes straight through the column. And keep in mind here that the overall mechanism used here is chromatography, and that the salt controls the elution. Now let's contrast that with pH gradient elution. Here, a change in the protein charge disrupts the protein to solid phase interactions. So proteins will elute from the column when the pH reaches a point where they have no charge. And this is an important difference from salt gradients, as they will not interact with the rest of the column. This affects the column length used, the loadability, and sample matrix effects. Typically, pH gradient methods can use shorter columns, also have shorter run times, and just as importantly, lower salt concentrations. And the mechanism here is mainly trap and elute, and the physical properties of the column are less critical. Basically, it's isoelectric focusing rather than chromatography. Buffers and pH control are required, but it's less prone to the difficulties and error that we saw when we were discussing salt gradient methods. Okay, and so now taking a closer look at pH gradient analysis. What effect does the column length actually have? Well, resolution usually increases with column length in standard chromatography. On this slide, we're showing chromatograms for 3 different column lengths, from 50 millimeters to 250 millimeters, done using pH gradient analysis. It's clear there's little to no difference in resolution with column length when using a pH gradient. As I mentioned, the mechanism is trap and elute, so the rapid on-off mechanism gives no further interaction with the column once the protein is off the initial binding site on the column. And this is an important point. It means that you can use shorter columns without compromise, which has the possibility to bring charge variant analysis into the world of high throughput analysis with extremely fast runs. And this can be speeded up dramatically by using a shorter small particle size column. And on the slide you can see now, we show the separation of 5 therapeutic bio-therapeutic monoclonal antibodies with initial runtimes for charge variant analysis of about 10 minutes. And this was done using our MAPPAC SCX10 column. But with some pH gradient optimization, each run can be taken to below 2.5 minutes without a loss in resolution. That's quite a significant reduction in analysis time. And actually, just for reference, if you're thinking about how long that optimization work might take, the total optimization time for these 5 antibodies here was just half a day. And on screen now is an example of a more complicated monoclonal antibody with multiple variants. You can see chromatograms before and after treatment with CPB. Some of the later eluting peaks are lysine truncation variants. You can easily see that these have been removed following the CPB treatment in the lower trace. The retention time RSD values are also very good, as you can see from the table that's just underneath the chromatograms. And on the right, you can now see an overlay of 10 different runs. It might not look like it, but there really are 10 runs there. And this method is so reproducible that if a difference occurs from one run to the next, for example, if you're sample screening, then that can be very easily observed. Okay, we're going to change gear slightly now and take a very quick look at the use of LCMS. So we've already discussed the use of salt and pH gradient analysis. Mass spectrometry adds an additional dimension of characterization, particularly when used with powerful deconvolution software. So here's a mass spectrometry example, the analysis of a monoclonal antibody under both native and denaturing conditions. Under denaturing conditions, the upper trace that you can see on screen, the protein is unfolded and the charges are fully exposed. And so the charge envelope has a mass to charge ratio of between about 2,500 and 4,000. And it's obvious that there's far more structural information here than with just the chromatogram, with all of the glycoforms being clearly visible. So now if you contrast that with native conditions shown in the lower screen, in the lower trace, the protein remains folded so that only the exposed surface charges are available. This means that the charge envelope is smaller, and obviously a consequence is the M to Z range is higher. So here at around M to Z of 6,000, due to obviously there being less surface charge on the folded protein. Now this does give better spatial resolution compared to the denatured analysis. But I guess the most important thing, the most significant takeaway from this slide is that both of these states give additional and complementary information about the structure of the protein. And in an ideal world, it would be best to use both denatured and native analysis. However, an issue with the use of mass spectrometry is the control of the pH using buffers. To run LCMS, the buffers must be volatile and clean enough to use with MS. In fact, mass spectrometry can be a rather demanding technique, and I'm sure there's a number of you in this webinar who are aware of that. This does make the CX1 buffers we've been discussing incompatible, and so we have to look at other buffers, some of which are shown on screen. And these need to be at low concentration to avoid ion suppression in the mass spectrometry. And unfortunately, there is also a buffer gap at pH 7 to pH 8, where it's very hard to find suitable buffers. So when we use LCMS, we're essentially adding a third factor, the mass spectrometer. So the choice of column, choice of buffer, and requirements of the mass spectrometer are all related and interdependent. So it's your job to reach a compromise with those three interdependent factors. So the mass spectrometer requires low salt concentrations and volatile buffers, and the mass spectrometer conditions do need careful optimization. You'll want the buffer to control the pH across all of the required pH range, yet its concentration will need to be low for best mass spectrometer sensitivity. And the buffers will no longer be strong enough to control the pH well, so the column should have low buffering capacity and allow easier pH control with a weaker buffering system. A MAPPAC SCX 2x50mm column is suitable as it is a strong exchanger, short in length, and can be used at a higher flow rate to speed up the pH equilibration. Next, we show an example of the difficulty and method optimization for charge variant mass spectrometry. The column here is 250 millimeters long. And if you've been listening carefully, you probably would think that's too long. And indeed it is. This column is probably 5 times too long. It has 5 times the buffering capacity of a 50 millimeter column, yet no increase in resolution. So this causes problems in equilibration times, and there are huge changes in the monitored pH that doesn't correspond to the programmed gradient. So the next run starts before the required pH is even reached. The ammonium acetate buffer A has a low pH. To get this to pH 10 requires titration with almost a 10 times excess of ammonium hydroxide. So the ammonium concentration is close to 500 millimolar. This is also the counter ion in cation exchange, so a salt gradient is actually being performed and with high salt. And the ammonium acetate does not buffer as expected, giving much higher actual concentration, causing yet more equilibration problems. So, although this method would give you a result, it would be very difficult to reproduce from column to column. And here's a better example, this time using the MAPAG SCX 50 millimeter column and a buffer system made up of acetate, carbonate, and ammonium hydroxide. This time, the monitored pH does track with a programmed gradient. I'm going to switch over now to briefly show you the mass spectrometer data. This shows excellent results that can be achieved with a well-designed method, the glycoforms being readily identified and the mass accuracy also being very good. The low salt concentration in both buffers also results in better sensitivity. And this method is easily transferable and does not require pH titration. If you're interested, this work was done in conjunction with NIBIT, which is the National Institute for Bioprocessing Research and Training, based in Dublin. And this has been published, so this data is readily available if you want to check out the method or indeed to have a look at the data. That same fast method development is also possible with charge variant mass spectrometry, and just as it was with the CX1 UV method that we showed a few slides back. Here, a method was established within 3 chromatographic injections by stepwise customization of the gradients applied while keeping the total gradient time to under 10 minutes. Again, the optimum buffer for this UV method are our CX1 buffers, as this system gives much better pH control. And remember, I know I've said it a few times, always be sure the measured pH values actually match the calculated ones. Okay, so we're at the summary slide. What have we covered and what have we learned? Both salt gradient and pH gradient ion exchange chromatography are powerful tools for charge variant analysis. I think you'll agree the choice of buffer is extremely important, particularly with high salt loads, and this can have a negative effect on the pH. Because of this, we highly recommend pH and conductivity detection, particularly during method optimization. And specifically for pH gradient elution, short columns and very short runtimes can be achieved without compromising the resolution. This really can be a high throughput technique. And we've also taken a quick look at adding mass spectrometry to get additional insight into structural information about your protein, with the caveat that you need to carefully balance the choice of buffer and column to best optimize the mass spectrometer performance. Now, just a quick mention about the products and the solutions. You'll be pleased to know, and probably not very surprised to hear, that Thermo Scientific supports a wide range of workflow solutions, from peptide analysis and subunit analysis to intact protein analysis and aggregate screening. And naturally, those workflow solutions include the column chemistries too. For protein and peptide separations, we have the ProPak, ProSwift, PepSwift, AccuCore, and Acclaim columns, covering a range of chemistries and particle and pore sizes to get the best possible resolution. We also have specific columns for monoclonal antibody-based proteins and peptides, including ion exchange, size exclusion, protein A, hydrophobic interaction, and reversed phase chemistries. For glycan analysis, we have optimized Hillic wax and also Hillic RP mixed mode columns. And for oligonucleotides, we have the DNA pack range. It is a huge range, and the newest additions to this portfolio are the MAPAC RP for intact protein isoform and subunit separations, and also the DNA-PAC RP for oligonucleotide separations.